This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, the podcast all about viruses. I'm Vincent Racaniello, and this is episode number 363. Recorded on November 13th, 2015. I didn't know it was 13th, Friday the 13th. Wow. Joining me today from Western Massachusetts, Alan Dove.
Good to be here.
Do you know it was Friday the 13th?
I did, and in fact, my wife had a minor car accident this morning.
Is everything okay?
The car's met up and at the shop, and we're annoyed about it and all that. But yeah, everybody, no injuries, and everybody was all right. But it was just a really annoying incident.
Yeah, car things are annoying.
Yeah,
and time consuming and costly, and
so coincidentally, when she texted me about it, I was sitting at the tire shop having a tire fixed on my car, and reading the papers for today's episode.
Sitting at the tire shop reading the papers.
Yes.
Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody.
What's the weather like out there?
It's kind of gray, and let's see, the temperature, 42 degrees, kind of drizzly, but it's not, no, it's kind of dried up, actually. But today's 11/13/15.
Yes.
Oh.
So that's kind of cool.
It is.
Yeah.
Here it's,
and I forgot to mention it's 58 Fahrenheit and a little blustery here.
See, right now it's 13 Celsius on Friday the 13th.
Oh, wow. It's 14 C here.
And we got nice clouds and a sunny sky. It's been very mild here.
Oh, yeah.
Yeah. Very, very mild. And it's already the second week in November. Also joining us today from North Central Florida, Rich Condit.
Howdy, everybody. How you doing?
Are you happy to be stuck in one place for a while?
Well, I'm leaving Tuesday for Austin. Okay. But yeah, I've been happy to be here for a couple of weeks or whatever it is. I don't even know how long it's been. It may be as much as a month or 6 weeks, I guess. But yeah, we're going to hit the road again pretty soon.
Good. Is it hot down there?
No, summer is finally over. This is great. Now we're into the good stuff. It is 76 degrees Fahrenheit, which is 24 C. It is not a cloud in the sky. The humidity is 40%. Dew point of 51. Tonight is probably going to be in the low 50s. It is just absolutely gorgeous. Thank God we're finally here. So there you go.
Yeah, up here we don't say these things. We like summer, but down there,
yeah. Summer gets. I've gotten used to this, so summer's okay. But this year, for some reason, I think I was just in a sort of a crabby mood anyway. But it got tedious. I got tired of it. But this is all good. This is great.
This episode of TWiV is brought to you by the Department of Microbiology at the Icahn School of Medicine at Mount Sinai here in New York City. They had such great results last year with our ad series. Every student had heard of the program through TWiV. They got great students, so they're back. Mount Sinai, of course, that's where I got my PhD. Great place.
Mount Sinai got the TWiV bump.
Mount Sinai got the TWiV bump. Composed of over 20 virology laboratories centralized in one building in the heart of New York City. A perfect fit for anybody who's interested in doing research on viruses. Right now, they want to recruit graduate students. Deadline is coming up December 1st. Now, most people have probably already got their applications in, but if you are thinking of graduate school and you haven't applied to Mount Sinai, you should do so. You have time. It's a terrific program with all kinds of viruses, and you can check it out at mssm.edu/MIC. K-E-Y. No K-E-Y. Just MIC. I can't help it. I grew up with M-I-C-K-A-E-Y, yeah.
Right.
M-O-U-S-E.
Exactly.
There you go.
Yeah, I heard that they just found a new lost black and white Mickey Mouse cartoon.
I didn't know that.
Let's go.
Isn't that cool? How can you lose it? Where would it be? I guess in...
storage room somewhere.
just like smallpox, right? In a closet?
Right, yes.
Yeah.
Anyway, you should check out Mount Sinai. We're really grateful for their support. And, nobody else has ever taken us up on this. You other programs, what's the issue? Why can't you advertise on TWiV? Don't you want great graduate students?
And now the experiment's been done, and it works. So.
It works. Everywhere I go, the students know of TWiV and get those on. We have undergraduates listening to us, right? Yeah.
Yep.
Yeah.
So,
and Caleb's going to be applying to graduate programs eventually, right?
Pretty soon.
Yep.
Pretty soon. So Mount Sinai, that's where, let's see, so Anna Sesma, Adolfo Garcia Sastre, Peter Palazzi, the chairman and the guy who I worked for. Oh, those are all off the top of my head. Oh,
Matt Evans.
Ben
TWiVer.
These are things we're pulling out off the top of our heads. And really a bunch of really cool viruses to work on there. And it's New York. New York is great.
Yep.
Chris Basler, Ben Hurley, right? Gene Lim.
Yeah.
Gene Lim, who I think used to be in Florida, Rich, is that right?
No, she interviewed here.
She interviewed there, right?
Yeah, she came, I think, pretty close to coming down here, but went to New York instead.
And she wrote in a long time ago to TWiV a few times about dengue. And we can't forget to mention Domenico Tortorella, because I want to say his name. Love it. Tortorella. Anybody with two L's in their name, they have to be good. All right, Mount Sinai, thank you for your support of TWiV. I want to shout out to Oregon Health and Sciences University in Portland, in the state of Oregon. Okay, I said it right. Oregon. Not Oregon, but Oregon.
Okay, it's the gun part. Right.
But if I got it wrong, guys, tell me. I just visited, had a great visit. I was invited by the graduate students to do a keynote at their retreat, which was a little bit, I guess it was in Portland, but a little bit on the edge, up on a hill. It was called the World Forest Center. And we have an email which came in from one of those students, and she warned me, there are trees and plants here, so you might not like it. But anyway, I had a great time, and I had dinner with a bunch of students, so I want to shout out to them. So L'Oreal actually picked me up at the airport holding a sheet of paper with a picture of polio on it instead of my name. I loved it. You
mean polio the virus.
Polio the virus.
An iron lung, right?
No, polio the virus. I saw it right away. I said, that's got to be me. And also Erin, who initially invited me over a year ago, Rebecca, Sarah, Lisi, Ashley, they all went to dinner with me. And I'll put a link to photos that I've posted in Facebook of the faculty.
These names are all female.
They're all women, yeah.
That's great.
Cool.
Yep. And we're at dinner, and the guy goes, I'll serve the women first. And I said, who else is here? So I had a great time. Really nice place, nice people, good science, good basic science. Molecular biology, really, really good. Now, you guys may remember this paper we did from Lou Picker's lab about the simian cytomegalovirus vectored SIV vaccine, right?
Sounds familiar.
I'm talking to my TWiV colleagues here. Yes, well, there's a lot of translational research going on there, but this meeting was mostly basic stuff. Viruses, bacteria, parasites, really good. So I'm glad to see basic research flourishing in the Pacific Northwest. So thanks, guys, for that visit. Cool. It was really fun.
Last time I was in Portland, we visited I think I told you about this experimental rose garden, which is probably done for the season by now, but it was amazing. It's a place where growers who have developed new strains or whatever of roses plant patches of roses to sort of test them out with the public and out in the environment and that kind of stuff. And there's just thousands and thousands of these things. It was spectacular. Nice town, Portland. I like it.
Very nice. And I had breakfast with our friend Ken Stedman. You guys remember him?
Yeah, I saw the picture of that. That's great.
Yep.
So he gave me his card. It says, Professor Kenneth Stedman, PhD, extreme virologist. Portland State University. Nice. Isn't that great? All right. Next thing up on our agenda, we have a Facebook posting from Ken Folta, right? Who put this up? Rich Condit? Was this?
I found
it.
Want to tell us about that?
Simply, now we've talked about Folta before, and it's not Ken, it's Kevin Folta.
Sorry.
We've talked about, look, the comments are gone.
No, they're all here.
Oh, they're all there. Okay. So we've talked about it before. Kevin Folta is a professor at UF in the Department of Plant Pathology, I think, who has spent a lot of time over the past few years trying to communicate to the lay public the real science behind genetic engineering of plants and genetically modified organisms. And so he speaks at various events, and he also has a podcast and stuff, and he got in trouble with the anti-GMO crowd because of a blog that originally appeared in PLOS Biology, was it? That used Freedom of Information Act emails and acquired emails and basically misrepresented his activities as being a shill for Monsanto, bottom line. And so he took a whole bunch of flack, and he's fought the good fight, but now posts on this Facebook post, "Hi everybody, I'll keep it short. The attacks are relentless. I'm under a lot of pressure on many fronts. I'm taking the opportunity to disappear from the public visibly and focus on my lab and students. It's been a challenging time. I appreciate the support. I'm grateful for your wishes, but this battle is vicious and one-sided, and I think I'm well served bowing out of the public science conversation for the foreseeable future. Thank you."
Well,
I mean, there were threats on his family and stuff like that. It was bad.
Well, so this came up during my visit because I talked in my seminar a lot about communicating, and someone stood up and said, I think I mentioned the Folta issue, and someone got up and said, the anti-GMO people, they have lots of money. The anti-vaxxers seem to have lots of money, but he gets a little bit of money and he gets hell for it. So what are we supposed to do? How can we fight this fight? It's a good question, right?
Yes.
Yeah.
And I said, well, I don't have a good answer, but, you have to be careful where you take your money. I prefer if our voices were funded by our listeners. And she said, well, how do you know you're going to have enough money? I said, I don't. We just have to see.
Right. Right, exactly. I mean, the issue with the funding is that the anti-GMO folks and the anti-vaccine folks are not government supported, and so they don't have to disclose anything.
Yeah.
So the actual support for the anti-GMO movement, certainly this has been documented, large quantities of money flows in from the organic food industry, which is a multi-billion-dollar operation that has a huge vested interest in demonizing GMO plants. And we don't know exactly how much of that money is flowing into, but we do know that there are some rather high-profile examples where they've funded lobbying campaigns and this sort of thing against, drumming up fear. And the anti-vaccine movement, they've got some very high-profile stupid people who have gotten money through other means, and they provide some, and I don't know where the rest of it comes from. But none of that has to be disclosed, whereas somebody like Folta, who's a publicly funded researcher, is susceptible to this sort of really abuse of the Freedom of Information Act, and that's what led to this disclosure of an email. And yes, he did take some money from Monsanto, not directly, but it went to something he was involved in, and if you are inclined to paint that as a conflict, you could. And so that is the problem, that he got involved with something that could be construed as a conflict, even though it's not.
So, Alan, isn't it true that the appearance of a conflict is just as bad as having a conflict? In
a case like this, yes.
Yeah.
Yes. Because this is not something, nobody's filing a lawsuit about this, so it doesn't... If it went to court, it would be, no, there's no conflict. You can't prove a conflict of interest here. But that's not what's going on. The court here is the hype, and in the court of hype, the appearance is everything.
Yeah. Well, it's too bad he has to give up his traveling, his podcasting.
It's really unfortunate.
Even his fake podcasting. Yes.
I mean, he made some poor choices in terms of going into a debate that was this high profile. But I don't think he did. Yeah, he certainly didn't do anything unethical. He just did some things that, if he had understood how his opposition actually operates, he probably would not have done these things.
Right. All right, we have a couple of snippets. The first sent in from Justin, who writes, FDA approves first of its kind product for the treatment of melanoma. And this is a virus, a herpes virus.
Yes.
Being used to treat melanoma. Now, this is the first oncolytic virus approved in the U.S., is that correct?
That's correct.
Yes.
Because one is in use in China, I believe, for neck tumors, but this is the first in the U.S. And I love the name of this herpes virus, Talimogene laherparapivec. Did I get it right?
I think so.
Somebody must sit around for months trying to figure out a name that nobody could possibly pronounce or remember.
Well, there's actually a whole. You're kind of right. There are rooms full of people who sit around at drug companies coming up with drug names, and the constraints on it are really rather severe. You have to come up with a name that hasn't been used, that is in some way descriptive to physicians, at least, and that's that's going to be accepted by the FDA, and that's not there. It's not too promotional in the name, and it's very hard to come up with drug names, which is why they come up with these awful ones.
The only sense I can make out of this one is it looks like recombinant virus vectors of one sort or another are going to be called VEC.
Yes.
Mm-hmm. Yeah, that's
okay. But the rest of the name.
Herp indicates herpes virus.
Right.
Oh, there's a herp in there. La herp.
Yeah.
Yes.
Just abbreviated TVEC.
Yeah.
Oh, they tell you how to pronounce it here on Wikipedia. Talimogene laherparapivec. I did it almost right. There was a great blog post, which I will link to, about how drugs are named, the actual reasons for it. I can't find it, but we used it when we wrote a chapter in our textbook on this. I will post that. So this is a very modified herpes virus, which has a number of modifications. It's got a deletion of a gene called ICP34.5, which does not allow it, which prevents it from infecting non-tumor cells, so it makes it tumor selective. There's a deletion of ICP47 gene, which is an inhibitor of antigen presentation, so infected cells won't be cleared by our T cells. And then there's the insertion of a human GM-CSF gene, which makes a protein that recruits dendritic cells to the tumor site. So the virus gets into the tumor specifically, it's lysing them, and then it's producing GM-CSF, and that recruits your own immune cells to the area. So this has gone through all the phases. It just finished the phase 3, and that's after that it was approved by FDA very recently.
Yep.
This is a really nasty disease. The patients in this trial, and there are 436 patients used, where they had inoperable melanoma, right? So last resort. And, some of them got the vector and some of them got nothing, or maybe GM-CSF alone. So I guess you can't not treat them with anything, right? So the current treatment is GM-CSF. So it's either GM-CSF or TVEC. And they measured a response rate, which is, the tumor doesn't grow for so many months. But and so this was approved based on a very favorable response rate in the TVEC arm of the trial. But I was just looking at the survival, right?
Doesn't change.
Really marginally, right? So the untreated people died after about 18.9 months. So that's it's an average. There's a range there. And then TVEC, 23.3 months, right? So we still have a way to go, obviously.
Yeah. By the way,
this is administered by injecting it directly into the tumors?
I believe so, yeah.
And with melanoma, that's, you're not going to get everything doing that.
Yeah.
Right.
Because, these trials are done with people who have failed everything else.
Yes.
Yeah.
So they're in pretty serious shape. So it could be that you're going to see something a little different now that it's licensed and it can be used on people, I presume, in other stages of the disease.
Right? Yeah, earlier times, right, where you might have better treatment response. Yeah,
right.
Exactly.
Right. So the Talimogene laherparapivec, by the way, I guess is what you would call the generic name. Imlygic is the name they're going. IMLYGIC.
I was going to ask whether it's imlygic or imlygic, because we don't know how to pronounce that either.
Well, it's a lytic. So I'm thinking imlygic, but it's lysis, so it could be imlygic.
You know, they could have left out that last vowel.
Yeah.
It would have been a real internet name then, imlygic.
Yeah.
I think it's worth pointing out in general the principle of how these oncolytic viruses are working. They do two things. They actually kill tumor cells, but in the process, it stimulates the immune system and exposes the immune system to a lot of tumor antigens, some of which maybe the immune system doesn't ordinarily see. And so in addition to killing the cells, it's stimulating the anti-tumor immune response, and those things both work together. And generally, I go back to a TWiV by Pat Moore a long time ago that was one of the most informative TWiVs that I've participated in, where he pointed out that the pathways that are involved in growth regulation overlap significantly the pathways that are involved in fighting off viruses. So cancer cells that lack growth control are also quite often defective in antiviral defenses, and that generally is why a lot of these virus vectors grow better in cancer cells than normal cells. So you don't have to target them necessarily in the same way people thought they would have to be targeted. They grow better on cancer cells than normal cells. So it's oncolysis directly and stimulation of the immune response by an agent that grows better in cancer cells than normal cells.
Right.
There are also some other approaches using for other tumors where the vectors have a gene that converts a prodrug into a drug, right? And and that it makes it more specific for the tumor than being everywhere. So then you treat these people with both the virus and the prodrug, and the prodrug gets converted in the tumor, and it's killing that directly. So this is really evolving very quickly, this field, and we'll see more.
I found the Wikipedia page on this vector pretty informative. Be good to put that in the show notes.
Yep.
And I went back to the original article and looked at that last night, where they show the percent surviving cells of various melanoma tumor cells that they have. And there's, anywhere between 4 to 5% of the cells, depending on the time they look and which cell line and vector they look at. So it's not that the virus vector is killing all the cells.
Oh, yeah, sure. And
just as another thing, this is in gene therapy, and I happen to think just now, I wonder when this came out. 2003 was when this came out. So that just gives you an idea of how long drug development takes.
The first mention of this vector, you mean, right?
Right.
Yeah. Mm-hmm. 12 years.
Yeah.
That's clinical trials take a long time.
Yeah.
They did
fairly typical for something like this, I think.
Yeah. Mm-hmm. It's unfortunate. And the field has gone a long way in 12 years, too.
Yes. The bad news is you have cancer. The good news is we can give you herpes for it.
Yeah.
Right. All right, we have another snippet sent in by Stephanie, our guest, a couple of episodes ago. Can I offer a snippet for today? I have an influenza paper to share, PLOS Pathogens, entitled Influenza Transmission in the Mother-Infant Dyad Leads to Severe Disease, Mammary Gland Infection, and Pathogenesis by Regulating Host Responses. Right up my alley. To be honest, if this had been posted before my TWiV, this would have been the paper I would have chosen. Some interesting takeaways: bidirectional transmission of flu from infant mother and mother-infant via respiratory and mammary route. 2 mammary gland cells from ferrets and breast cells from humans were permissible to influenza. May I just say, Stephanie, that should be susceptible and permissive, not permissible, because you got both susceptibles, receptors, and permissive is inside. 3, the study also suggests that influenza that can be transmitted to infant ferrets suggests that flu can be transmitted to infants through the mammary gland. However, this study will use direct inoculation of the mammary gland to prove this. Since it is not the mammary gland that becomes inoculated with influenza first, I remain skeptical that in natural infection, mammary gland alone would cause the severity of disease they saw. However, I definitely think the mammary gland could contribute. But then neutralizing antibodies and other neutralizing factors in milk could prevent virus transmission, particularly at low titers. Regardless, the mere fact that the mammary gland could contribute to infection is alarming and must be considered in disease pathogenesis. I believe that studying virus transmission via milk is imperative to human health. We must understand this mechanism. The HIV milk transmission model is well established. However, there is such a paucity of info for other viruses. This is a paper in PLOS Pathogens, as Stephanie noted. The first author is Stéphane Paquette, and the last author is Alison Kelvin. And this comes from Canada, Spain, China, and
Italy.
Italy's in there. I just get blind to Italy, I guess. Italy, yeah, it's a multi-country study. And it uses a ferret model for mother-infant dyad, and they infect them with influenza. So this is kind of a new thing. We've talked about ferrets a lot before as good models for flu, but here we have mothers with nursing babies, and you can infect either one and see who gets infected.
And the combination of the mother and the baby are the dyad, not to be confused with a dryad, which is a forest nymph.
A forest nymph?
Yes.
It's a triad, really?
A dryad, yes.
Oh, dryad is the forest nymph.
Right.
I love the word dyad. It's a great word. If any of you have watched the television show called, I forgot the name.
I haven't seen that one.
I forgot. It was my pick of the week months ago. It's about cloning people, and there's one actress plays like 7 different characters. Anyway, the name of the nasty company in that show is Dyad. You know, if we were live and we had a chat room, people could tell us right off the bat.
That's right.
It's a motivation to have a chat room.
Right.
Well, in this paper, they do a series of infections of either the mother or the ferret with 2009 H1N1, the pandemic.
The mother or the infant.
What did I say?
The mother or the ferret. They're both ferrets.
The things you say.
Yes.
Actually, in this study, they had humans nursing ferrets.
No.
Oh, God.
What's wrong? You have a problem with ferrets? They have sharp teeth. Yes, mother ferrets and baby ferrets. And they can inoculate either one intranasally, or as we'll see, in the breast directly. And they used the 2001 H1N1 pandemic strain, which apparently causes particularly nasty disease in pregnant women and babies. They wanted to see if they could get any clues about that. So if you first do the experiment where you inoculate the baby with flu, you do intranasal inoculation into the 4-week-old ferret, and then you put the ferret with the mother, and they nurse and so forth. They both get sick. The baby gets sick, and then the mother gets sick, and they can recover virus from the nasal cavity, nasal wash, and there's pathology in both, in the upper and lower respiratory tract. So I presume what's happening there is, the mother and the baby are very close, so there's some contact or inhalation transmission of virus, even though the baby is nursing, but I think it's probably a respiratory route.
Right. That part I didn't find very surprising.
No. I mean, that makes perfect sense, right?
Right.
And that's a very realistic sort of experiment. They cite this factoid in the discussion that infants and toddlers are infected with 8 to 10 respiratory viruses a year, which is a totally believable number if you've had kids.
So you could imagine in a family, maybe you have a new baby, and then the father may infect the baby with flu, and then the baby is nursing, and then it gives it to the mother, right? So that can happen. Then they do the other way. They infect the mother, and then they put the babies with them, and they nurse. And then the mother gets flu, and so does the baby. All right? So that all makes perfect sense. I don't think there's anything unusual there. They see virus in the respiratory tract. There's pathology. It's typical influenza. The next one they do is they have they have an experiment where they've inoculated the baby. Again, we're going back to the baby ferret with influenza virus. And then they look in the mammary gland, and they find virus in the mammary gland, both infectious virus and RT-PCR RNA. They also find it in the nipples. So the baby is nursing and presumably transferring the virus to the mother, and it apparently can get into the mammary gland, and it replicates there, and the glands shed virus in the milk. So this is new as far as I know. I don't think anybody's done that. I don't even think they've even looked in people at this. The replication of the virus in the mammary gland is accompanied by destruction of the architecture of the gland.
And by the way, no matter how you're reading this paper, whether you're reading it digitally or in a printout, because I was reading it printed out at the tire shop, and from comments you all have made before we started recording, this isn't any better in the digital edition. They use the same pattern of illustrations for figures 1, 3, and 5.
Yeah, so you can't tell which is which.
So you flip through and you see this figure, and then you're flipping through and you see this figure again and say, wait, I just saw this figure. No, it's different. This is the one where they infected the mothers. Yeah, oh, no, this is the one where they infected, so.
I was doing this on an airplane on a laptop, and I was going crazy. Because as it is, there's not much air in there,
And one of them, the first one like that, figure 3, doesn't have figure 3 written anywhere on that piece of paper. The entire legend is on a different page.
Yeah.
So I had to go in and handwrite all the figure numbers on it.
Yes, the legend, when you print this out, PLOS is primarily a digital journal, but they try to do a nice job when it prints out. It looks very pretty in the page layout. It's just that things are spread out over too many pages.
And what's the point of having that whole column of blank space?
Right. In our next paper, we're going to complain about what they did to compress this.
You can't win. You can't
they
win.
make these figures too small, and in this one, they make the figures too big, and they have all this white space that you could take copious notes in if you wanted to, I suppose.
I think the white space is kind of it's a style, right?
Design element, but it wastes paper.
I think the white space is probably an artifact of this being primarily an online paper.
Well, if you stretched out the print to be the whole page, it would be really wide, and you don't want to do that.
You'd have to do it in columns
then, yeah.
But then the page layout and the web page layout would be very different, and you'd have to maintain both those layouts. And so
I do prefer columns, though.
I think I do too.
Anyway, I think at minimum they should try and put the legend on the same page of the figure, right? So some of these figures could be shrunk.
Yeah, they could, or they could make that huge margin of white space smaller in order to get it on. Yeah, whatever.
Well, PLOS, if anyone's listening from PLOS, probably not, but they won't listen to us.
McFadden is listening.
But he probably doesn't have control over the typesetters.
McFadden has control over everything.
That's really nice.
Well, but I think this is a PLOS-wide thing. It's not just PLOS pathogens. I think it's
I mean, these guys are all in cahoots with each other.
All right.
Okay, so
Grant.
if Grant hears us complain loud enough, he'll say something.
Yeah.
All right. He's hearing us.
Grant, you're listening, right?
At the same time, they then ask in these infected mammary glands what genes are going up, what genes are going down. So they do. Wait, what page am I on now? I lost myself.
16 of 34.
Oh my gosh!
Well, it's figure 7.
All right, so they did RNA sequencing, I think here. Yeah, global gene expression analysis, and then they say what genes go up and down. And it's very interesting that for me, what I thought cool was there are decreases in genes involved in milk production, which is pretty neat.
And didn't they say at some point that in fact, under some circumstances, milk production is actually compromised in this situation? Right. Is that right?
Yeah, there is a subsequent experiment where they actually do that. Right. So there are lots of changes in the expression in the breast caused by infection, and they list them all. But I think the most interesting is the one involved in milk production, which correlates with their finding in the next experiment that in fact milk production is decreased. So they actually inoculate in the next experiment. They inoculate the mammary gland with virus through the lactiferous ducts. All right, and then. Yeah, the virus, they do this and they put the baby back with the mother, and both of them develop influenza. So presumably, we know the virus is replicating in the mammary gland. It presumably spreads to the infant that way, right? And then, I guess...
So you don't necessarily know that. If the mother gets sick as well, it could still be respiratory.
Right.
So that raises the question of when you inoculate just the mammary gland, how is the mother infected? It must be by some respiratory route, because I don't think it's getting in the blood and spreading to the lung and crawling up on the apical surface of the cells.
Wouldn't it be interesting if the infant gets infected and then infects the mother?
So
there is a time course here, right, of this?
Yeah. So in this experiment, the infant gets infected first, I believe. Yes, that's correct. The infant is infected first, and then it goes to the mother. So I presume this is all aerosol-based, because during breastfeeding or when a baby is with the mother, you get aerosols, close contact and aerosols. But this shows directly that if you put virus in there, it replicates, and then it can come back out again and spread to a nursing infant. So if you have twins and one of them has flu and you're nursing both, you could spread flu to both of them this way, right?
Right. There's this terminology, retrograde flux.
Retrograde flux, right.
Yes.
Yeah. It describes
I see.
fluid.
So, viral load in the nasal wash. The infant gets sick first, and then the mother. Okay.
So in this experiment, they looked and found that milk production was decreased, right? Mm-hmm. And so later on, they talk about how, if you are nursing a child and you're infected in the mammary gland, this could impact milk production and the ability to nurse and so forth. So that could be an outcome of replication in the gland. That's basically the paper, right?
So all the way through this, I was trying to figure out, the novelty here is that you show that it is shown that a nursing mother can have virus in the mammary gland.
Right.
Okay, and that virus will apparently grow in some of the mammary cells. They do we didn't cover that, some cell culture experiments where they show that cultured cells from cell lines from mammary glands will grow virus. So, and the implication is that there can be transmission both directions, from the infant to the mother, or that you can get transmission from the infant to the mother's mammary gland eventually, and transmission the other way around, and that can be a source of infection. But it's really hard to prove because you can't do the appropriate controls. Okay, I was trying to think of controls for this, where you would, have a non-lactating female or a male that's been infected with virus or something. I don't know, but there's an interesting line in the discussion.
I have it because I'm just going to talk about it.
Go for it.
I'm pretty sure it was put in for reviewers or people just like you, because in fact there's a large part of it that's italicized. Controlling the experiment through other means, such as, italics, the addition of a non-lactating adult to the cage of mother and infants, brings ethical considerations, as the presence of another adult would put the infant ferrets in danger.
So I guess if you put anybody in there other than mom, they'll eat the babies.
Yeah.
Yes.
Same with mice, yeah.
Yeah.
Yeah. So if, here's the thing, if let's say a mother acquires respiratory influenza, I don't think that is going to go to the breast, okay? Because the virus typically is not viremic.
Right.
Mm-hmm.
However, they do point out that the 2009 H1N1 was a bit unusual, and that in some patients, they could detect virus in the blood. So, I don't know, in some virus strains, maybe it could reach the mammary gland and then replicate there and spread. Who knows? But I don't know if we can ever do that experiment, right? So I guess you could take a ferret and just infect the mother and then see if virus appears in the milk without having a baby in there, right?
Yeah.
You could do that.
Mm-hmm.
And the converse experiment, now let's say you have a baby who's infected, the mother is not, and we know from this that the breast can be infected. Would the mother get infected if you could somehow block respiratory transmission? Would it go into the blood, into the lung? I think that's even less likely, but...
Make her wear a little N95 mask.
Yeah.
That's right. That would be doing it. Yes. Yeah, they got to make them for ferrets.
One thing that I wanted to comment on too was that in the figure 5, where they gave the virus to the kits through the nose, that's where they measured viral RNA in the infant feces. Right. And so, does that counteract our thoughts about, in mammals, influenza not being a gut virus?
This is just virus that swallowed, the babies they're eating virus and milk and they're passing it through. You can see that,
dose,
because it doesn't replicate there. But in that same figure, you can see the bottom panel is virus in the blood and there's nothing there.
All right, yeah.
So that just shows that it's not getting in there.
Right.
Anyway, this is kind of relevant to the next paper because you may be wondering, when an infant is drinking milk, mother's milk, how would it get infected with influenza virus if it's just swallowing this, right? Well, as we'll see in the next paper, the soft palate is an important part of flu replication, and that's in the back of your mouth.
Mm-hmm.
Yep.
You're going to do this education advertisement?
Thank you, Kathy. This episode is also sponsored by ASM Education. Grad students, postdocs, and early career scientists are invited to apply to the ASM, that's the American Society for Microbiology, ASM Scientific Writing and Publishing Webinar Series. This is a 3-month, 6-part program. It takes place from January to March of 2016. My, the years are moving on, aren't they? I'm doing something which involves 2017. Oh my gosh. So ASM runs a variety of educational programs like this. They're very useful for you. In this particular program, you're going to learn from ASM journal editors all about the scientific writing and publishing process. They will talk to you about writing titles, abstracts, figures, legends, and the whole manuscript review process. The deadline for applying is December 1st, a couple of weeks off, so you do have time. Go to the website, bit.ly/SWPIONLINE15. If you don't hear me, go to the website, TWiV.tv. We'll have a link there. We thank ASM for their support of TWiV. Our main paper for today is a paper which we first heard the results of at ASV this summer, Kathy and I and Rich Condit, right?
Mm-hmm. Yep.
You guys all heard this? You were at the talk, right?
Mm-hmm. Yep.
Presented by Kanta Subaro, who I had invited to speak in the morning session. Tremendous influenza virus researcher at the NIH. Title is, and I remember at the meeting one night she said, it was accepted. And this paper, right? Mm-hmm. And it's accepted. It's in Nature, and it's a letter. It's called The Soft Palate is an Important Site of Adaptation for Transmissible Influenza Viruses. Yep. First author is Seema Lakdawala, who I met at ASV and took a selfie with, because as Kathy knows, that's what I do, right?
Mm-hmm. Yes.
Kathy dances, I do selfies. And it's on Facebook. We'll put a link to that as well. Well, this is all about influenza virus transmission, which we've talked about maybe as much as Ebola virus, right?
Maybe.
On this podcast. And what you need to be transmitted. And of course, one of the models that people use is a ferret model to measure transmission. You put ferrets in adjacent cages. In a lab, you infect one intranasally, and you see if the other gets infected. You can measure virus in nasal washes. Just by putting a little buffer into the nasal cavity. I guess you put it in the nose, right? And it goes into the nasal cavity and sloshes around. Then you pull it out, and if there's virus there, you can measure it by plaque assay if you're really cool, or by PCR if you're less cool. Hey, by the way, on my visit to Oregon, I saw lots of plaque assays. I was very happy.
Excellent.
People came up to me, said, come and look at my poster. I have plaque assays on it. It's just great. And then one guy at breakfast, one of the faculty, said, I'm going to build a tower of plaque assays in my lab, in my office too. We're making a trend here. It's cool. All right, back to the paper. As everyone may know, if you've listened to our TWiVs, receptor binding is an important determinant of transmission. And let me give you a brief review of receptor interactions in influenza virus. Influenza viruses bind to a glycoprotein on cells. And the most important part of that glycoprotein is the very last sugar attached to the protein called sialic acid. It's a single 6-carbon sugar. Sialic acid binds into the HA, and then that sialic acid is attached to other sugars and then to a protein, and that helps bring the virus into a cell. Now, the next sugar... Do
glycolipids work as well?
I don't know if glycolipids work. That's a good question.
Because you can have sialic acid on glycolipids, right?
Of course. You could. I don't know. You were measuring the limits of my ignorance.
Well,
can anyone Google it while we talk? Sialolipid influenza virus.
Sure.
So the first sugar is sialic acid. Then it's linked to a bunch of others. And the second one, third, fourth, fifth. But the link between the sialic acid and the second one is apparently very important for host range and transmissibility. So for humans, human influenza viruses, the ones that infect us all the time, they like to bind to sialic acid, where the link to the second sugar is called alpha-2,6. The 2 and the 6 carbons are linked together, covalently joined. And avian viruses, which typically don't infect us very well, like to bind to alpha-2,3 linked sialic acid. So this just refers to the link between the first and the second sugar. All right, so remember that alpha-2,6 is human, alpha-2,3 is avian, and presumably for an avian virus to become a pandemic human strain, would have to, at minimum, recognize alpha-2,6 linked sialic acids.
Right, and that's some of what we've talked about with flu host range in the past, and papers that have been done on this have looked at exactly this problem. How does an avian adapted virus change its sialic acid preference or its receptor binding preference in order to make it into mammals?
Now, in the ferret studies that we have talked about with transmission, they take an H5 virus, which is an avian virus, binds alpha-2,3. They make it bind alpha-2,6 by changing the sequence of the HA. And then they pass it in ferrets, because that's not enough to get aerosol transmission. Then they passage it in ferrets by aerosol, and then they get other changes in the HA that allow it to be transmitted. So what they did...
Okay.
Yes, sir.
Okay. Time out.
So what do you got?
This is a gain of function experiment, isn't it?
No, the one I just talked about is, we knew that was a gain of function. This one in this paper is actually a loss of function.
Yes.
Yeah, but I mean, the one you just talked about is gain of function.
Yeah,
when I first saw this paper, I wondered, so is this now legal?
Well, so the ferret experiments I'm talking about, the Kawaoka and the Fouché, those are certainly gain of function. Yeah, they selected for the ability to transmit by aerosol among ferrets, right?
Right.
So here they start with a human virus that transmits already very effectively by aerosol among ferrets. Right? The 2009 H1N1 pandemic virus, which actually was the subject of the previous paper as well.
Right.
This nicely recognizes alpha-2,6, right? And it transmits very well in ferrets. So what they said is, let's change it to recognize alpha-2,3 sialic acids and see what happens in ferrets in the transmission experiments, right? So they can make 4 amino acid changes in the receptor binding site of the HA, and this changes it to recognize alpha-2,3.
But now it's not transmissible.
Well, it is. When you do a transmission experiment, it is transmitted, but there's a delay in transmission.
Right, because it selects for a change.
And apparently this is a change that's selected for, right.
Okay. So this is a rescue of function experiment.
Okay, fine.
Well,
and that change, in fact, is to change one of the original 4 guys, right?
Exactly. Now,
it reverts.
They made a loss of function, and then the ferret did the gain of function, so humans are not responsible.
Right.
This should be permitted because animals are not subject to human law.
Okay.
All right.
All right.
Yeah, so originally one of the 4 amino acid changes is D-aspartic acid to glycine at position 222, D222G. Okay, so now the virus that is transmitted from one ferret to another, they deep sequence this RNA, and they find a mixed population in virus pulled out of the nasal cavity, right? And in particular, this position, 222, is mixed. The airborne contact ferrets, the mixed population was in the original infected ferret.
Right.
Mm-hmm. And then the one that received the virus by aerosol, they have only the revertant, which is now called G222D, to make you more confused. The original was D222G, and now
we've gone back.
We've gone back. It's completely reverted.
I really had to work on that one.
And the inoculum that they had put into the original ferret was okay. It didn't have a mixture. It just had D222G. All right, so this is...
So since it's a reversion, is it a reverse genetics experiment?
No, it's just genetics.
It's just genetics, yes.
It's just genetics. This amino acid change has been seen before in natural isolates. And people have thought that it makes the HA receptor binding site better bind to alpha-2,6 linked sialic acids, but not altering alpha-2,3 linked interactions. Okay, so these are done with modeling experiments. In other words, so far we have this reversion of this single position 222, and theoretically it's thought that improves alpha-2,6 binding. So remember, we've changed the binding of this virus to make it alpha-2,3, right? which ferrets don't have, so it shouldn't transmit well. But this change is selected for very rapidly. So they do some glycan binding studies. So they have synthetic sugars with sialic acids with different linkages to the second sugar, and they stick those on a plastic dish, and they ask, does virus stick to them? Does virus bind to them? It's kind of like Rich said in our pre-show discussion. It's sort of an ELISA because they detect the virus stuck to the sugar, stuck to the plate, using an antibody to the virus. Right? Now, we all have a little confusion about the actual execution of this experiment, which I tried to clarify by emailing the first author. But like a good scientist, she's working at the lab and she's not answering her email at this moment, and that's fine. The conclusion is, I will tell you the conclusion of this experiment, and then we can talk about why we don't understand it. The mutation, the amino acid change G22D, doesn't change alpha-2,3 binding, but increases binding to alpha-2,6. Okay.
Mm-hmm. Right.
So
and we're gonna take the author's word on that, because otherwise we'd have to decipher figure 2.
So remember, alpha-2,3, the virus was changed to recognize alpha-2,3.
Right.
Now the G22D makes it bind much better to alpha-2,6.
But it can
if
still
I
bind
understand it correctly,
alpha-2,3.
not any old alpha-2,6, but long-chain alpha-2,6.
Yes.
Right?
Right.
Guys that have more sugars between the glycoprotein and the sialic acid, so they're longer.
Right.
Right.
So there's no decrease in the alpha-2,3, but just an increase in the alpha-2,6. And this is the virus, again, the mutation selected for in the ferret in this airborne experiment.
Right.
Do you guys want to explain the problems that we have with this?
Well, just that in the second part of the figure where they show this glycan binding, it seems like they're not showing the control, or it's a binding signal percentage, and it's a percentage of something. And so they say that the mutation caused no change in the alpha-2,3 linked sialic acid binding, but substantially increased binding to long-chain alpha-2,6 linked sialic acids. But I don't know how to see that from the data.
Yeah, you need something to compare it to, right?
Right.
Yeah, it's not clear what 100% is or what they're comparing it to. The appropriate control, it seems to me, would be against the original virus D222G. Okay, because they're saying that it's that the change back to the reversion back to G222D causes an increase in the binding to the alpha-2,6 long chain sialic acid guys, but I don't see where the control is.
Yeah.
Right. And the different titers across the x-axis don't enlighten me either. But
right. Yeah, and by the way, if you print this on a black and white laser printer and you're sitting in the tire shop trying to read it, it is completely impossible because they've used multiple colors in the graph, and all you see is these shades of gray. And and to decrypt it, you also have to go to the methods section, which of course is not in the main body of the text.
Are there 50 shades of gray?
I don't know exactly.
Or 256 most likely.
Probably 256, yes.
Yeah.
At any rate, we'll take their word for it.
We will
The
take their word.
change increases the binding to the alpha-2,6 long chain sialic acid dudes.
Right.
I have a feeling nature said, you don't need this, and they threw it out. But we'll see what they say. Now, their conclusion is, this is very important, we now demonstrate conclusively that airborne transmission requires gain of long-chain alpha-2,6 linked sialic acid binding, and contrary to previous suggestions, loss of alpha-2,3 linked sialic acid binding is not necessary. And that statement, number 4, is a paper published in 2007 by Tumpy et al., where they said you had to lose alpha-2,3, but you don't have to, apparently, at least for this virus, right?
Right.
Now, all right, so this amino acid change is selected for in the ferret, and it allows transmission. Where does it happen? Where does the selection occur? It's a good question that maybe not everybody would have examined, right? Exactly where in the ferret? Is it in the upper tract, the middle, the lower tract?
Right.
So they look at different parts, tissue sections and samples from upper and lower tract, and what they find, and they look at the virus to see where it is changing.
And just a little history on this. It's there are different sugars in different parts of the respiratory tract, and that actually is an issue with respiratory viruses. Some that bind deeper in the lung bind, something, and then some that bind shallower in the nasal cavity bind a different sugar. So you would be concerned about this.
Yep. There's a great figure which I have in my lectures, which has a human with alpha 2/3 or alpha 2/6 in different places throughout the tract. Actually, the eye has alpha 2/3. So avian strains could theoretically infect the eye, and you could then select for 2/6. And there have been some outbreaks of eye infections on bird farms with avian influenza viruses in that very same way. They find that, surprisingly, the viral RNA from the soft palate is enriched. They say remarkably and uniquely enriched for G222D. Yep. I like that phrase.
Well, I have a little quibble with the uniquely on day one, because in the nasal wash you see some of that, in the soft palate, in the nasal turbinate. I mean, you see a little bit of that revertin coming up in all those things on day one.
But it's uniquely high levels of it in the soft palate, I think, is what they're getting at.
Well, in one
not necessarily
individual,
at day one, but subsequent days.
Right. But if they say it on day one,
right.
So on day one, there's one that was 80%, but in the nasal wash, there's one. It's just how they phrased it,
Right.
Yeah.
I would have taken out the uniquely. Remarkable.
But if you back off and kind of look at this at a distance, it's clear that the soft palate is behaving differently than the other tissues, in particular at days 3, 5, and 7.
Yeah.
And this got probably be a good
mostly
place
the revertin
to insert our anatomy discussion about where the soft palate is, right?
I'm looking at this cross section of the head. I too looked up to figure out make sure I knew what the soft palate was.
Yes.
Look at the tongue.
Yeah.
I didn't realize it was such a massive organ sitting in there.
Yeah, go to the butcher and look at some cow tongues sometime.
Listen, my mother used to make me eat that crap. Oh, God.
Well, it was presumably sliced up, right?
No, she would bring it out intact
and
slice it in front of our eyes. I think deliberately to gross us out.
But you became a biologist. Probably nothing grosses you out now.
It's got this layer of epidermis. It's got the taste buds and the whole thing. Jesus, it's awful. It's just awful.
Yeah,
God.
Memories. But yeah, they always draw the tongue looking like a clam, rearing up there. I guess that's the way it is, right?
I thought of it that way, yeah. It's a huge organ.
It is.
Well, so this shows the palate, but the hard palate's in front and the soft palate's in the back.
Exactly.
That's correct.
So where's the soft palate? The little
soft spot in the back of your throat.
So the part that says oral cavity behind that arrow?
No, kind of up above that.
Right
I think
above
the lighter thing that has a little dividing line.
So
if you take that whole stippled thing that is labeled palate and draw a vertical line about halfway down, the dorsal part of that is the soft palate, and in front of that there's bone.
You could put your finger in the top of your mouth and start feeling, and when it stops being hard, the bone, it becomes soft.
All right, let me try that. I'm
about to puke.
I'm gonna throw up now.
You shouldn't.
It's soft way in the back. Yeah.
Okay, here you go. I got
so
another.
So the soft palate is partly in your mouth, then, right?
Yes.
Yeah, and that's there's a supplemental figure that shows it for the ferret.
Oh yeah.
I'm pretty sure.
There you go. Have a look at that one.
Oh, it's not actually in the nasopharynx then.
No, it contacts the nasopharynx up above and it contacts the oral cavity below.
Yeah, that was news to me.
Was it a little flappy thing? It looks like flappy, but it is, yeah?
Yeah.
Huh. So, this paper where the ferrets get infected by drinking milk, I was wondering if one mechanism could be, gets into the soft palate.
Could be.
It could be, right?
Yeah. Mm-hmm.
Sure.
Huh. So I wrote that yesterday, and then this morning I said, nah, it's not right. Because I was thinking that hard thing was the palate, but it's not. That's a hard palate, yeah.
So they call it I presume they call it the hard palate because that's actually bone.
Yeah, it's bone in
there. And that part of it looks like it does not have tissue that's part of that contacts the oropharynx. It's just the oral cavity.
Oh, no, I think it does.
Think so?
Yeah.
Oh, maybe so.
Yeah,
or
at least the nasal part of the oropharynx.
Right.
I mean, it's in contact both sides.
But I think it's really interesting that the soft palate has, epidermis contacting both sides. And that clearly they're different in terms of the distribution of these sugars. We should be recording this.
Well, yes, it is, and I'm probably going to splice it in at some point because it's very useful because we're not going to repeat it. So it looks like, so that normally people do nasal wash to measure flu replication. So you're basically getting a measure of replication in what's labeled as the nasal cavity here, and all the epithelium lining that, right?
Right, in the top figure, yeah.
Okay,
and you would collect stuff from the nasopharyngeal surface of the soft palate. Doing that, I presume.
Right. Sure. I mean, it's there. It's part of it. Yeah. But you wouldn't know specifically there versus the rest of the place. I mean, that's the point of the paper, right?
Right.
In part.
Right. And in this cross section that I just put in here, these flappy things, are those the turbinates?
Yeah, the ridgy things, right?
Yeah.
Yes, those are turbinates, right. And I'll never forget, Peter Palazzi always used to talk about the ferret as having turbinates like people. He said one of the few animals that have similar turbinates, which he thought was important for flu. And, it's in the nasal cavity, so it's probably replicating on the surfaces, right?
So are the olfactory receptors on the turbinates?
No.
Or all over, or where? Do you know?
They are on the cribiform plate.
Oh, my God, you know that. Where's that? I have to look that up.
I have another. I think it's towards the back.
You'll have to look at your cribiform sheet to figure that out.
I think it's on the top and towards the back. Yeah, look up
cribiform what?
Plate. There's one reason I know that, because people used to think polio entered that way, because the olfactory receptors, of course, go right through the bone. The nerve goes right through the bone into the brain. So it's a wonderful passageway. And many people said this is the way poliovirus gets in. And some of the early trials, prevention trials, were to coat people's noses with aluminum salts.
Right.
Which did nothing but irritate their noses.
Well, didn't some of those trials also permanently destroy their sense of smell?
Yeah. Mm-hmm.
So it looks like the cribiform plate is at the very back end of the nasal cavity, right? As you say, right up against the brain.
Right. I've got it against one of these illustrations is showing it against the top.
Right. Okay.
With the olfactory nerves coming in from there.
So in the top drawing that I've put in, the original one here, where the guy is facing left, where would the olfactory, the cribiform be? Over oral cavity, where the letters are in that general area?
Approximately in there, and I think maybe up into the area over palate.
Okay, so they say this revertant is selected in the ferret soft palate. The ferret selection in the ferret soft palate. I don't know. That sounds like a title to me. We'll see. All right. When, so is this enrichment responsible for infection? That's the question, right? So they do an interesting experiment where they do, again, this transmission study, but they only expose the uninfected animals for 2 days. So you're looking very early after infection of the original ferret. Everyone got that? So you infect your ferret,
right,
and then you put the others in the next cage for 2 days, and you take them away. Mm-hmm. Nevertheless, 2 contact animals and 3 out of 4 airborne animals seroconverted. So 2 airborne contact animals shed virus, and 3 out of 4 airborne animals seroconverted. So they get infected anyway. It's transmitted even only after 2 days, and they sequence the RNA from the contact animals, and they have the G22D virus. So this selection happens very quickly, and they say it's possibly originating in the soft palate, because you can't really prove it, right? You can say the soft palate does select quickly and the transmission occurs quickly. You put the 2 together and you say this is probably what's happening, but
you need ferrets that don't have a soft palate.
I was just going to say maybe there's a genetic alteration you could make that would,
Well, the best you could do is somehow knock out the alpha-2,6 long chain sialic acid
maybe, yeah.
Glycoproteins or conjugates from the ferret. Good luck with that.
Yeah.
Right,
Yeah.
you'd have to be making transgenic ferrets.
At this meeting in Oregon, I saw someone with a poster where they had made mice with no lymph system.
Ooh.
There's a transgene you can put in that prevents the production of lymph capillaries or something.
And how are those mice feeling?
I don't know. I didn't ask.
I imagine not well at all.
I don't know if they live a very long time, but they're looking at immune responses. Okay, where are these sugars? Can we learn something by staining these different parts of the oral and nasopharynx? So they stain the soft palate with lectins, where you can say we can look for alpha-2,6 or alpha-2,3 sialic acids, and they see that you can find alpha-2,6 in the ciliated respiratory epithelium. So there's the cells on the top of the soft palate exposed to the lumen, let's say, and the mucus-secreting goblet cells, which would also be on the top there. They could also find alpha-2,3 linked sialic acids in the connective tissue underneath the epithelium and in serous cells of the submucosal glands. So these are beneath the epithelial cells, and I suppose not likely to be infected, right?
Right.
Yeah, I don't know.
Right.
Yeah.
The epithelium is the place. So there's a lot of alpha-2,6 there. So, if you put a virus in, that's alpha-2,3. But as we know, RNA viruses are the masters of mutation. So probably a little bit of virus with alpha-2,6 binding capability, even though you can't detect it by sequence. And that's rapidly amplified, right?
Yeah.
And my read on this is that the most enriched portion of the soft palate, the portion of the soft palate that is most enriched for the alpha-2,6 long-chain sialic acid conjugates is in the nasopharyngeal portion of the soft palate, which makes sense because it's, I guess anything would make sense, but that's a good, physically a good place for spreading the virus from nasal secretions.
Well, I guess you're going to inhale aerosols with virus, right? And then it would go back there and replicate on that target.
Back out in the same direction.
Yeah.
That's a really cool figure in the extended data figures. Figure 6 shows that.
They also, so they find that long-chain alpha-2,6 are higher in the soft palate than the trachea and the lungs of the ferrets anyway, right?
So I don't know if we said this, but one of the really novelties of this is that prior to this, nobody's really looked at soft palate.
That's right.
As a place for virus replication or virus attachment. And so since nobody's looked, they haven't seen this, and so this really reveals that.
They also make an interesting comment, even though the turbinates, which are the kind of ripply surfaces in the nasal cavity, the turbinates in the olfactory epithelium have high levels of long-chain alpha-2,6. They're not enriched for the mutant virus. It's only the palate. So they don't find the selection going on up in the other parts of the nasal cavity, which is really curious, right? I don't know why that would be that way, but we'll see someday.
Yeah.
They also looked in human tissues. They got soft palate of humans and pigs, and they found long-chain alpha-2,6 linked sialic acids on the respiratory epithelium and goblet cells of the soft palate of both. So in the end, humans are pigs. Yes. So we have alpha-2,6 on our soft palate, which raises the possibility that we may select for alpha-2,6 binding viruses in a similar way. Anyway, so that's basically the story, right? Anything else?
So bottom line, you don't have to lose alpha-2,3 binding.
Right.
And the replication in the soft palate is important.
Mm-hmm.
Right.
Yes.
At least, and they say at least for this one virus, right? Because it is one virus, so people should look at some others in the future. But it's a very nice study, and it was fun to hear about it at ASV. And I said when I heard it, we have to do this on TWiV. So there you have it.
Mm-hmm.
The soft palate.
Cool paper.
It's very cool paper.
Yeah.
Okay. Do I have another ad, Kathy?
Nope. I think we're right into email.
Not till later. All right. Some email. This one is from L'Oreal, who I met out in Oregon. And I asked her the origin of her name, and she said that her parents were hippies. Anyone understand? What is L'Oreal? It's some kind of mythical thing?
I
don't know.
No? All right. Hello, TWiVers. I'm a fifth-year graduate student at Oregon Health and Science University in the Molecular Microbiology and Immunology Department. And first, I'd like to say thank you to me for accepting our invitation to be the keynote speaker at our upcoming department retreat. We're very excited for your visit and talk, although I'm sorry to have to tell you that the retreat is taking place at the World Forestry Center, where you will be forced to look at plants all day. Should have read this last week.
How bad was that?
It was all inside. I didn't see many plants. It was very nice. I don't mind all plants. It's just certain ones.
Yeah.
Why do people think I hate all plants? I guess
you were pretty adamant about the cherry trees.
Yeah.
I don't like the cherry trees, but I don't want to have house plants either. That's ridiculous. And unfortunately, my house has got a lot of house plants.
House plants are good for you.
Yeah, they make oxygen, right?
Yeah.
And I learned
they send out vibes too, Vincent.
They do.
Okay. Yeah. They calm your spirit.
Look, I never liked dogs, right? And that my son has 2 dogs, and now
right now everybody's just wondering about the kind of houseplants Rich is growing down there.
Yeah, right in Florida.
They calm your spirit. Okay, dude.
Anyway, the dogs now. After 8 years, I really like these dogs. They calm my spirit. When I come home, they actually come to meet me.
See?
Nobody else does that.
Yeah.
They come and they wag their tails. I think that's pretty neat that dogs like humans.
Yeah.
Continuing with L'Oreal's letter, I also wanted to say thank you for the interview with Dr. High, episode 350. My current work focuses on adeno-associated virus as a vector for gene therapy, and it makes me happy whenever I hear a shout-out on TWiV about how successful this little virus has been. Which brings me to a point I wanted to add to the ongoing gain-of-function discussion. And I apologize if this was mentioned before and I missed it, but I find it funny that the main focus in the AAV field right now is all about gain-of-function. The last American gene therapy meeting was filled with talks about using different techniques to create new AAV vectors with novel properties, mostly focused on infecting specific tissue types or avoiding neutralizing antibodies. A standard protocol in the field is to create a library of different AAV capsid mutants, point mutants, chimeras, peptide insertions, etc., and do directed evolution or biopanning to isolate mutants that have a higher tropism for the cell type you want to target in vivo. And she sent a paper, a review, Directed Evolution of Novel Adeno-Associated Viruses for Therapeutic Gene Delivery, which covers all this. I'd imagine that if someone tried this with flu, the backlash would be harsh. I guess the ban on gain of function doesn't apply to AAV since it's still considered non-pathogenic, and most people are already antibody positive for it. Remember we did that Kawaoka paper a few weeks ago where he tried to get flu to grow better in eggs, and people went nuts over that.
Yeah.
Now, if you came up with an AAV that used something like the CRISPR system to delete an essential gene in humans, I don't know, maybe.
I don't know, maybe.
There's probably some gain of function that would freak somebody out there.
Speaking of how common AAV is in the population, I think Dr. R will be interested to know that AAV has been proposed as a commensal virus of mammals. AAV is known to lower titers of its pathogenic helper viruses in culture, and epidemiological evidence implicates AAV as a negative risk factor for developing cervical cancer from HPV infection. She sends along a review by Flott and Ken Burns, a good friend of Rich Condit's.
My goodness.
Down there in Florida, right?
Yep.
It's called adeno-associated virus, a ubiquitous commensal of mammals. We call it a commensal.
This is back from 2005.
It is. And in this they talk about how, lots of people are infected, but maybe it's beneficial, as L'Oreal says. Thank you all so much for the Twix. In my lab, we're pro-virus all the time, trying to infect people and bypass the immune system. So I really enjoy learning about the other side of virology. Yeah, that being where we try to prevent infection, right? Pro-virus all the time. That's a good title. Twix, pro-virus all the time. I love everything about your podcast, from the great journal article discussions to the ongoing conversations about the plight of postdocs and gain-of-function studies to the weather. Right now in Portland, it's 63F, 17C, sunny with scattered clouds, winds 2 miles per hour. I'd say it's a beautiful fall day, except this year has been our warmest and driest we've had for a long time. The changing weather is alarming, especially for those of us with urban gardens. At least all of our peppers did well this year. Thank you, L'Oreal.
Now, the problem with being pro-virus all the time is you'd never get out of the cell.
Right.
Wonderful. Dude. Did you think of that in the tire shop?
I just came up with that now as this email.
This selfie you have with L'Oreal makes it look like you've got a polio virus earring or something. I guess it's an adenovirus earring, maybe.
Yeah, she had a couple hanging on her poster. And you can read the title. She had an AAV poster there. And she had plaque assays and electron micrographs, really nice stuff. And L'Oreal is going to do a postdoc with Jude Samulski.
Excellent.
At UNC. Continue AAV. So, what did you say, Alan? The trouble with being pro-virus is that you're in the cell all the time.
Yeah.
Yes.
L'Oreal is just a variation on laurel, which is comes from the bay or laurel plant. It's one of the lesser known variants of laurel.
And we have a selfie, as Rich said. And if you look at this selfie, you will see that L'Oreal is a Padawan.
A what?
A Padawan.
What's that?
The braid?
Yes.
Is that what? Okay.
And presumably when she defends, she will cut it off. Because that's what Padawans do.
That's what the Jedi do, right?
The Jedi do. Okay. Alan Dove, can you take the next one, please?
Okay. Karen writes, as we move into flu season and the fear of vaccines again ramps up, I have a question that has come up in general conversation and I don't know how to answer. If flu mist is a live attenuated virus that sheds, though it's not a danger to immuno-intact people, could it pose a risk to the immunocompromised? For example, we generally avoid getting the flu mist if we're going to have contact with those isolated in a hospital and undergoing immunosuppression before transplant. But is it safe to be around those who are out in the general public and undergoing regular chemotherapy for cancer treatment? Another example given was someone taking immunosuppressants for sarcoidosis. Any thoughts or expertise you could share for us lay people out here fighting the good fight for vaccines would be greatly appreciated. Please excuse any inaccuracies in my questions. I'm not an expert, just an enthusiast. And don't worry about the inaccuracies in your question. There weren't any. It was just a legitimate question. And Vincent has provided a very handy link to, I guess this is MedImmune's site.
Yep.
Which it's a myth versus fact, which is not the most effective educational format, but they do outline exactly that question about immunocompromised people. I guess it's the third one down.
Yeah.
And the upshot is, if people are capable of being out and about, walking around in the public with all the nasty things that come with, that they're exposed to with that, then they're certainly okay to be around somebody who got flu mist.
Yeah. It's no problem. There's no reported transmission to immunocompromised or immunosuppressed people. Yeah.
And they do, just I think out of an abundance of caution, they do recommend that for people who are in isolation, that those around them not be given flu mist.
They also cite the really high odds of possibility of reversion to wild type influenza. So it's not likely that that's going to be an issue either.
Right. You're not going to get the flu from flu mist. That was kind of the focus of much of their approval process.
There has been some studies, I believe mostly done in daycare centers by the developers of flu mist, that show that there is a small possibility of the vaccine virus being transmitted from one person to another. It's in about 1 or 2% of vaccinees in a situation like that where people are under crowded conditions. But even then, you're passing vaccine virus, so there's really no big threat to that.
Right.
Very well informed and entirely appropriate question.
Yes.
Okay, because it is a live virus vaccine, so those are all appropriate questions, and the answer basically is there's essentially no concern.
All right.
This is a good segue into our little communication we had by email before the show, where I got a communication from our, because I'm on the hospital email list that says that if you haven't been vaccinated for flu this season and you're going to be working in the hospital, you got to wear a mask.
Yep.
Yeah, for us, it's not even if you're working in the hospital. It's if you're going to the cafeteria. Yeah,
Wow.
Pretty sure I'm not sure where they draw the line, but basically they came down really hard, and everybody, even undergraduates in my lab that would never venture off in that direction, had to show something, a proof of vaccination.
Wow.
The last phrase of Karen's, "I'm not an expert, just an enthusiast." That's like a Bones statement, right?
Mm-hmm.
Guy from Star Trek.
Right.
I'm a doctor, not an engineer.
Right.
Kathy, can you take the next one?
I lost track. Stephanie writes, "Good afternoon. I've appreciated your podcasts over the past years and wondered if you have an episode or article addressing the theory that the HIV outbreak in humans may have been facilitated by use of a polio vaccine derived from monkey kidneys. I've only ever heard the theory of HIV originating from some source of bushmeat and spreading rapidly through sexual contact in an urbanized setting to make its widespread debut on the world scene. Thank you for your work. Respectfully, Stephanie."
TWiV 300 is one of the occasions where this came up.
Yeah, did we talk about it a little on there?
Yes. So
we've talked about it extensively.
We've talked about it extensively on multiple episodes, I'm pretty sure. And this is based on a book that came out, probably almost 20 years ago now, by a guy named Edward Hooper, I think.
Yep, that's right. The River.
Yes. And Hooper was a journalist who went to extraordinary effort to try to prove his pet theory about the origin of HIV. And unfortunately, he really was kind of centered on trying to prove the theory rather than trying to test it. So he collected fragments and scraps of this and that and put it together into this book that made this long argument, which is really not consistent with the available evidence. So the upshot is that no, that is not what happened. We now actually, because of the book, people then went and did analyses, saying, well, okay, this is not consistent with the evidence at all. But let's just triple check, and it's now been conclusively proven that this is really not what happened.
Yeah, it's the phylogeny of the virus that's isolated from chimps in Africa by Beatrice Hahn and others clearly shows that it probably went from chimps to humans in the early 1900s, and the vaccine trials were done in the 50s.
Right, so, and there are a bunch of other issues with the theory behind the book. It just absolutely does not float. You can go back to, again, TWiV 300 is one occasion where we discussed this. I think if you search through earlier episodes, it probably came up in others too.
A cool thing, which I may have mentioned on 300, but I'll mention again because it's almost 100 ago. So the vaccines in question were made by Hillary Koprowski at the Wistar, and they were used in the Congo trials in the 50s.
Right.
And the idea was that they were made in chimps, which were chimp cells, which were contaminated with HIV or SIV, which then got into people. So they found some old stocks of Koprowski's vaccines at the Wistar and decided to look for, I believe they're looking for chimp mitochondrial DNA, just to see where the viruses had been produced and what kinds of cells, and also for HIV, of course. And I remember, I don't know how I got involved in this, but they asked me to aliquot the viruses and code them, and then they were sent to labs for analysis. So I met I think it's the president of the Wistar on the New Jersey Turnpike at a Dunkin' Donuts at exit something. And we got in the parking lot. We swapped this white foam ice box, right? He gave it to me. He didn't want to send it. So I drove down and he drove up. It was weird. He said, "It looks like we're exchanging drugs here." I said, "Yeah, but I'm not paying you anything." And
not
then,
the first time the Jersey Turnpike has seen an arrangement like this.
This is really funny. I took a picture of the vials, which I will put on the show notes because it's pretty cool. These old glass vials with paper labels on them. So I aliquoted them. I put them in aliquots. I coded them, and I met him again on the Turnpike a few days later, and I gave him the aliquots, and they tested them, and it did not prove. It showed that there is no HIV, and they weren't grown in chimp cells. That's been clear.
Right.
All right. We are down to Jackson. Is that correct?
Yes.
I believe so.
And that's Rich Condit.
Okay. Dear TWiVers, I was on Facebook and saw IFL Science's post about 4 types of anti-vaxxers and methods to change their minds about vaccines. I followed IFL Science links to the original article and thought that it would be an interesting article about anti-vaxxer attitudes and a nice addition to the paper in PNAS discussed in episode 349. Gives a link to this article. The article is open access and comes complete with tweets for attention spans too short for abstracts. Thanks for the great podcast. I love listening to them while I'm waiting for gels to run in the lab or in the gym. It's 54 degrees Fahrenheit, 12 C, 285 Kelvin, and drizzly here in College Park, Maryland, and gives also the IFL Science source. So this paper is by some behavioral psychologists, and it's actually their data, if you like, is mostly literature review of what other people have proposed. Mm-hmm. And this proposal of some categories of anti-vaxxers originates with the World Health Organization. And they've taken all that stuff and put it together and come up with their own 4 categories of anti-vaxxers, which is nicely summarized in the IFL Science Redux, I believe. I'm looking at this again. Yes. So the categories are complacency, convenience, confidence, and calculations. And they go through what these different categories are. People have different sort of conglomerations of reasons why they choose not to vaccinate, and then they investigate how one could talk to people in each of those categories to convince them otherwise. So it's an interesting...
It's a good paper.
And they have also suggested tweets that you can make to, like when people don't vaccinate because of complacency, communicate the risk of disease, that sort of thing, which you may not readily put together on your own. So it's very nice. And all the reasons why people do not vaccinate, which we've touched on briefly before, right? It's all here.
Just puts it in a nice package.
It's very nice. Thank you very much, Jackson. All right. This episode is also sponsored by ASM Biodefense and Emerging Diseases Research Meeting. Early bird registration rates for this meeting, it's 2016. They're going to end on January 14th, so you have a lot of time. This is a meeting originally conceived about biodefense, and then as biodefense became non-existent, I shouldn't say that, I don't know, but they expanded it to emerging diseases because not enough people were going or there wasn't enough talk. So now it's biodefense and emerging diseases. It's really a good meeting. I have been the last few years. They have great speakers like Tony Fauci and others. And I've done a podcast there in the last few years. And in fact, at this meeting, I will be doing one as well. And Rich Condit joined me a couple of years ago, right? Mm-hmm. We had Yoshi Kawaoka and your friend, the Pox guy, whose name I forget.
Dennis Ruby.
Dennis Ruby. It's a good meeting. It's in Arlington, Virginia. Oh, they're moving it out of D.C. this year. February 8th through 10th, the premier event to discuss how to defend against the growing threat of bioterrorism and shape the future biodefense research agenda. Yeah, it's easy to get to Arlington. There's a metro stop right there.
Yep. And no danger of cherry blossoms. It's, I think, too early.
February. Excellent. Might even be snowing, right?
Right.
I really love Arlington. What a wonderful place.
We had a couple of study sections there, right?
We did, and that's where Ed Niles lives.
That's right. It's very nice there. It's lovely.
I was there just a couple of weeks ago playing golf.
You've been, right, Kathy?
Mm-hmm.
Did you study section there? Yeah, that Hilton there right by the metro? Mm-hmm. Yeah, that's great. Very convenient.
I had an aunt who lived down there. A beautiful area.
Did you have an aunt also?
Yes. They have lots of aunts. But I only had one aunt there.
So Biodefense, ASM Biodefense and Emerging Diseases Research Meeting. You should go. asm.org/biodefense 2016. Very easy URL. We thank ASM for their support of TWiV. Let's now do some picks of the week. Alan Dove, what do you have for us?
I have an Ebola situation report of a sort.
Yeah.
So this is a YouTube video of a song called Bye Bye Ebola. And it's sung by an artist from Sierra Leone, I believe, and it features a lot of prominent people, including, I think, the president of the country and a number of other people dancing in it, celebrating the departure of this disease or the elimination of this disease from that country.
It's great.
It's absolutely fabulous. I love this video.
Yeah, I sent it to several people, with the subject line, "This will make your day." And the people are just dancing so great. And so, yeah.
It's just so jubilant and joyous. And yes, this is a major accomplishment.
Yeah, they have people gowned up too. It's pretty cool.
Yeah.
Mm-hmm.
They only got 26,000 hits. Come on.
Yeah, this really ought to do better than that.
Well, I just shared it on my Facebook page, so that'll go up to a couple million. Pretty. Yeah.
Oh, yeah, for sure.
It'll get the TWiV bump now.
You bet. Rich, what do you have?
So just a few days ago, the Google banner celebrated what would have been the 101st birthday of Hedy Lamarr. Were she still alive? She died at the age of 85, and I wondered, why is Google doing this? So I looked into it, and it turns out that Hedy Lamarr, I knew very little about her, except I knew the name and knew that she was a famous actress from like the 40s and 50s. But it turns out that she was also an amateur inventor and was co-inventor of a technology called spread spectrum and frequency hopping technology, which she, along with another guy whose name is George Antheil, who was a composer.
Yep.
They, during World War II, developed this technology as a means for counteracting radio jamming signals that might be a problem for radio frequency targeted torpedoes. In fact, they have a patent on it. The technology was never actually adopted by the Navy at the time. It was adopted later in the 60s, but its principles are incorporated into Wi-Fi and Bluetooth technology nowadays.
Amazing.
And
Yes.
Hedy was inducted into the National Inventors Hall of Fame in 2014. So I thought that was pretty interesting.
Hedy and George were thinking about this particular problem because it had come up in some conversation, and they started playing around on a piano and came up with the idea of sending notes of different pitches at the same time, like a chord, or hopping from one to the next. And they developed this scheme for frequency hopping. That's how Wi-Fi works. It's how Bluetooth works. And you can, because you're hopping through different frequencies, you can actually do extraordinarily robust encryption of radio communication this way.
Awesome. Kathy, there should be a Lego.
Yeah, I just, because I always forget which famous people were involved in this kind of thing, which famous women. And Julia Child was the other one I'd been thinking of.
Yep.
She worked out of
yes.
OSS headquarters in Washington
Right.
in research and development. One of her projects was a workable shark repellent used for downed flight crews and used for U.S. space missions with water landings. She also supervised a facility in China, handled countless top secret documents. So yeah, Julia Child, Marlene Dietrich, but I Googled female spies World War II and came up with this page with a bunch of different people.
Wow, she had 6 husbands.
Yeah,
Wow.
So I gave the best link here is a wiki link for Hedy Lamarr, and I read the whole biography, and there's significant sadness in her story, it's a real mixed bag. It's sort of, by the time I got to the end of it, I felt, kind of sad. So there are high points and there are definitely low points in her career. Very interesting person.
Wow, neat. All right, that's pretty cool. Sorry, who was that? Rich? Yeah, Kathy, you're next.
Okay, yeah. So I picked something that I've had on my list for a while. The link I gave is to phys.org. It's about an insect, plant-hopping insect, that has hind leg joints that are gears, mechanical gears. And so the headline is Functioning Mechanical Gears Seen in Nature for the First Time. And it's really cool. They show this pretty high image of the gears. And then there's also a little video that you can watch. And the paper was published in Science. I didn't go far enough back to check and see whether that was an open access kind of thing, but probably not. But you can get most of the information from this, and I actually saw it on another blog first. So it's I guess it's the genus is Issus or the species. I'm not sure. They call it ISSUS. And it's just really cool because they I'm trying to find the line here. It bears remarkable engineering resemblance to gears found on every bicycle and inside every car gearbox. Each gear tooth has a rounded corner that acts as a shock absorbing mechanism to keep the teeth from shearing off. And they lock together, ensuring synchronicity in leg movement. And it's just really cool. It's critical for how the insect can jump. Yeah.
Yes. Right, because if they don't synchronize the legs perfectly, then when they jump, because this particular insect jumps really, really hard and high, it would throw it off and it would go tumbling.
Yeah.
So, yes.
I think I remember this being on a doc before, and you pulled it for something else once, right?
It might be.
I seem to remember this paper coming out, and I'm just looking at the original paper, and it has even more images in it and a lot of stuff about how it works. It's really pretty cool. I can't tell if it's open access or not. I doubt it. Cool.
Thank you, Kathy. My pick is an article in Wired, which is called "Biologists Could Soon Resurrect Extinct Species, But Should They?" by Jacob Kushner. And it's a pretty neat story about, lots of species going extinct, and this is nothing new, but the white rhinos, for example, are just 4 left in the world, and they're not mating. They're getting old, but we do have their cells, and we can use technology and make pluripotent stem cells, and then make eggs and sperm and make new rhinos, maybe. Although I don't know where you'd put them to be.
Exactly.
Yeah, exactly.
To become a new rhino.
they're going extinct for a reason,
right?
And I don't like the reasons, and I don't like the extinction. But if you recreate them, yes, you say, Vincent, where are you going to put them?
People are just going to go out and shoot them again.
Yep.
Yeah. Anyway, there are apparently lots and lots of frozen animal and plant cells in many freezers throughout the world, and just right here in New York City, American Museum of Natural History has tons of them. And this article just talks about what would we do with them. So it's pretty neat. Check that out. We do have 2 listener picks. Jenny sent us the best listener pick ever, the people of Sierra Leone. It's okay. Celebrate being declared Ebola-free. Check out the serious dance moves, especially from Ebola Treatment Center staff.
Yes.
Yeah. Thanks for all you do, Jenny. I thought we could do it twice, because you got it first this week before Jenny's email.
Yeah. Alan was so excited about this.
I called dibs on
claim.
Yep.
Yeah.
He said there's an email. I got this one.
Right.
It's so funny. And we have one from Peter. Dear TWiV hosts, no time or ability to come up with a new creative way to address you. Oh, come on. It takes 5 minutes.
That's all right.
It's all right. edX course, Epidemics from Hong Kong University, might be of interest to your listeners. It's not too late since the course started on September 15th. The first graded quizzes are not due until November 24th. Enough time to catch up in case one is interested in a certificate. Week 1 features a panel discussion with Peter P.O. How was it said? P.O.?
We trip on it every time.
Yes.
Originally part of the first time this course run on edX last year. Speaking of MOOCs, is there any schedule of new iterations of how viruses cause disease and or Virology 1, how viruses work on Coursera? All the best from Incheon, Korea, where it is currently October 5th, 2 p.m. local, 22C, mostly sunny, humidity 33%, wind west-northwest 4 miles per hour, pressure 1,023 millibars, UV index 6, or in short, a perfect fall day. Peter.
He says MPS. Maybe that's meters per second?
Meters per second.
Meters per
Wind.
second.
Yeah.
Wind, yes. Yes, that's the metric wind speed measurement that's often used.
MOOCs. I don't know, Peter. I'm going to teach my course again next semester. I might MOOC it. We'll see. It's a lot of work, and I want to space it out a bit. But I want to try the edX platform, which is where the epidemics course that you're mentioning is on. It's interesting and different from Coursera, so I might do that. So stay tuned. All right, now, last thing I want to do here. I have too many copies of Principles of Virology, the new edition, so I want to have a contest to give some away. Yeah, the first time, we'll do it in several phases. The first thing is very easy. Follow us all on Twitter, and then write a tweet explaining why you listen to TWiV, and include the hashtag TWiV. All right, and the most creative entry will get a book set. This is a $190 value. I'm going to give it to you for free because I have 10 copies, and I can't store them anywhere. I only need one copy, and I have an electronic one anyway. So I decided to share it with our listeners. Okay, so I don't know if you guys all okay with giving your tweet?
Sure.
Yeah, I think so. Yep.
Okay, it's Alan Dove, Prof. VRR, Rich Condit, and Virus Prof. Sings. And you got to follow us all right? Because I want to hit 10,000 by the end of the year.
And then we have to keep track of it and let Vincent know.
And then do the hashtag, and we'll collect them all, and we'll decide.
Right.
We'll decide. When you tweet this, you don't have to have all of our Twitters in it, but you just put the hashtag in so we can find it.
Right.
I just realized I was not following Kathy.
Yeah. There you go.
Okay, fix that.
And be creative, and you can win a book. It's a 2-volume set, brand new, 4th edition. Still has plastic on it, wrapped in plastic. That's another movie reference. We'll see who can get that one. It's not actually a movie, it's a TV show, but that's close enough. Wrapped in plastic. You can find TWiV at iTunes, twiv.tv, and do send us your questions and comments to TWiV at twiv.tv. Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
By the way, both of our TWiV and TWiM from Ann Arbor are now released.
And pretty soon I'll be able to pick the video of Powell Kazanjian and the plaque ceremony. I just got word that they're available for us to look at.
So good.
That'll be soon. Yeah.
And then you're going to make that a pick of the week.
Yep.
Cool.
Definitely.
Very nice. Excellent. Rich Condit is at the University of Florida in Gainesville. Thank you, Rich.
Great fun. Thanks you.
And his Twitter is Rich Condit.
Yeah, I don't like all these cryptic Twitter things,
But Alan Dove is not cryptic.
Yeah, Alan Dove is not cryptic. There was this other, what is it, the real Poxdoc? We talked about that the other day.
Yeah.
That's really annoying. I don't know who that is.
Well, and the 2 fake Dixon DePalmier accounts, which that's just baffling.
Yeah, but they occasionally say nice things,
Yeah, they're not malicious from what we can tell.
Alan Dove is at turbidplaque.com. On Twitter, he is Alan Dove. Thank you, Alan.
Always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.
So I've been silent because I've taken me this long to get to the extended data figures. And I swore I saw a picture of the ferret nasal cavity and stuff, but now I'm not seeing it. There's a figure that shows the immunofluorescent staining. That's extended data figure 5. But here's an annoying thing. So there's extended data figures, and then there's supplementary information.
Yeah,
you can't download the whole thing at once.
It's ridiculous. These are out of control. Okay.
Yeah.
Why do you have to download it at all? Right? Why can't you just look at it all?
Now, why can't it just be there?
Yeah.
When you get paper online, it should just be there.
I had a lot of trouble with. So I read online. I don't print. I think you print out, right?
Print most of the time. Yeah.
And
What a dinosaur. Unbelievable.
This PLOS pathogens paper I had a lot of trouble with because the figures are huge. And I was on the plane on a laptop. You try and read a sentence, and then you have to go 4 pages forward to look at the figure. Then you forget where you were.
Yeah,
Wow.
I was thinking that it's that way on the paper, too. You had to go 4 pages forward, and each figure is a whole piece of paper, and then the legend is on the back side of it because of how it's printed out.
Well, with paper, you could kind of put 2 next to each other, I guess.
Well, eventually I had 3 piles on my dining room table. Here's the text, here's the stuff that's in between, and here's the figure that I'm looking at. And then when I had to turn a page, it was nuts.
We should probably record a TWiV about all this.
Yeah. With the PLOS pathogens paper, one of the annoying things that I found was that if I clicked on a figure to get the blow up, I could do that. But then when you close that down, it doesn't take you back to the same part in the paper. It takes you to the top of the paper, and you got to find where you were before.
Yeah,
I did find that if I opened all of the figures, there's an all of the figures link in a new window that I could put them side by side and go through the figures separately from the paper.
Yeah, it was actually the PLOS pathogens one that I had to have in 3 stacks on my dining room table. Yeah, it was crazy. Okay.
All right, let's do the rest of this.
And there's another TWiV.
I think I'm going to stick all that in somewhere.